Great. Welcome, everybody. We're going to get started in just a couple minutes. Give it until 7 Eastern, 4 Pacific time. Is it 6 o'clock Central time? 5 o'clock Mountain time? What time is it in France?
It's close to 1 a.m. in the morning.
Lovely.
You're a trooper.
Thank you, Xavier, for
Thank you.
being here.
Actually, I slept about three hours last night since I was stuck in Berlin because a flash retardation. So, yeah,
Wow.
it will be good when I will go to bed tonight.
Thank you so much for joining us and doing this presentation.
Okay.
So 1 a.m. We've got a variety of time zones. And for everybody joining, this will be recorded, so you can catch the recording later if you like. We'll send out an email in the next couple of days when the recording is available. Cool. See some familiar names. Michael, do you want to say hi to everybody?
Absolutely. My official hi, or are we
Well,
ready
you can say hi now and say hi again if you want, or we're pretty close to 7 Eastern. So
I was just
up to
going
you.
down through here to see how many folks we knew here at this point.
Yeah,
How
I
many
do see
people-
some names that I don't recognize.
How many people signed up tonight?
I think it was 83 or 86, somewhere around there.
I see Rachel's here. Rachel Cutchy from our PAB. Nice to see Rachel. That's great.
Jared's joined us from Annabelle's Challenge.
See Jared. Yeah, I haven't seen Jared in person. I think maybe since, well, maybe I've got to be 2 years.
Yeah, it's late for him too. So is it 12 a.m. for him maybe, or is it also 1 a.m. in the UK?
We'll see Jared at Science in Paris, I hope.
Oh yeah.
So
Okay,
why don't
well,
I do my hello? How about that, Katie?
sounds great.
So I'm the only person that stands between you and some extraordinary physicians tonight. Could not be more excited to be participating in this. It's the first first kind of seminar like this or webinar that I remember us doing, at least for some time. And I think it's a great opportunity to learn more about clinical trials and how one might participate. It's also a pleasure to be with the VEDS community. Great asset to the organization. Special thanks goes out really tonight to Peter Byers. Most people are probably not aware, but Peter initially introduced us to the VEDS movement, which it became, and we had a board retreat, and he suggested we invite Katie as well. And so not only did we meet Katie, we ended up adopting VEDS, and now we have the VEDS movement, and the rest is sort of history. So again, Peter, thank you for your leadership, and to everyone on this group, really, for your extraordinary care of VEDS and related conditions community. So I hope everybody has a wonderful night. I'm looking forward to the opportunity. And Katie, back to you.
Yeah, thank you, Michael, and thank you everybody for joining us tonight. If you don't know me, I'm Katie Wright. I'm the director of the VEDS Movement, a division of the Marfan Foundation. And today we're going to be talking about clinical trials. I'm really excited to have this presentation and opportunity to talk about clinical trials more in depth as we are approaching a couple for VEDS. It's just very exciting. So let's see. Okay, here we go. So tonight we're going to talk about the importance of research in VEDS, what it means to participate in a clinical trial. We'll go over some key terms that you might hear when we're talking about clinical trials, and we'll talk about analysis and outcomes. So we have a great group of speakers here tonight. We have Dr. Dietz, Dr. Xavier James, Dr. Peter Byers, and Dr. Shaine Morris. Grateful to all of you. As a reminder, there is a Q&A session at the end of this presentation. So if you have questions during this presentation, go ahead and put them in the Q&A box. The Q&A is going to really be focused about, around general clinical trial questions. So specific clinical trial questions really won't be answered in this webinar. Both of the clinical trials are still in the planning processes and not all of the protocols are finalized. So those questions might not be able to be answered for you tonight, but we will go over as much as we can, and we will have ample time at the end for a Q&A. So please remember to put your questions in the Q&A and not the chat box. And without further ado, I think I'm going to go ahead and hand it over to Dr. Hal Dietz, who's going to get us started today. And Hal, I'm going to hand you the remote control.
Thank you, Katie. It's really a pleasure to join you all tonight to talk about clinical trials in general, with a few early details about specific clinical trials in vascular Ehlers-Danlos syndrome. I am thrilled to be able to share with you some information about the purpose of this particular clinical trial, but also the hard work, the background, the legwork that goes into launching a clinical trial. Some of you might wonder whether clinical trials are done for broad general reasons or whether they're usually done for a specific purpose. And I think the latter is generally true, that a clinical trial is often or generally launched for a specific indication, for a specific condition. And in this circumstance, the specific condition is vascular Ehlers-Danlos syndrome. Clinical trials don't simply launch based upon a hunch, based upon a vague idea. They're usually based upon a lot of background, basic scientific research that points in a certain direction. That can be research that occurs using samples from people with a specific condition, such as cell lines or tissues. It can also be research that's based upon animal models of a specific condition. An animal model is simply a animal line that has been created to mimic or replicate the basic mechanism of a condition. And in this particular circumstance, animal models were created that involved the introduction of DNA changes, or what we call variants or mutations, that are known to cause vascular Ehlers-Danlos syndrome in people. In this circumstance, the specific DNA changes were introduced into the COL3A1 gene, one of the 20,000 genes in our body that specifically encodes for a protein that's called type 3 collagen. And in people with vascular EDS, a deficiency of type 3 collagen is the basic problem. Now, once you have evidence pointing in a certain direction, for example, in an animal model, it's important not to jump to the conclusion that the same treatment strategy will work in people. There's a principle in clinical trials that the investigators that are involved have to maintain what's called equipoise. Equipoise means that those people are equally receptive to the answer that the treatment strategy works, or the answer that the treatment strategy does not work. So that's the point where we're at now. There is evidence from animal models about a specific treatment strategy that we'll talk about. There is evidence in those animals that this treatment strategy can prevent important complications of vascular EDS, including vascular rupture. But we are at the stage where we're prepared to test that treatment strategy in people. Now, one thing we have to consider when planning a clinical trial is which people should be involved. And often that decision is based upon who is at risk for serious complications of the condition. We know that people in general with vascular EDS are at risk for vascular events that we'll talk about in a moment, and that those events can have serious consequences, including death. We know that the events that are serious in vascular EDS can begin in late adolescence and early adulthood. And therefore that flavored our decision to begin enrolling patients in a clinical trial that would extend into those age groups. It's also important to note that there are no current FDA-approved therapies for vascular EDS. We are respectful that many individuals and their physicians have used various treatment strategies in the past, and those treatment strategies will likely be respected and maintained in the clinical trial, but there will be more information that becomes available as the protocols mature and as they are finalized. So I'm having trouble advancing the slides. Katie, if you could advance the slide for me.
Yeah, sure, I can do that.
So as I mentioned, the beginning of the problem in vascular EDS is a DNA change in the COL3A1 gene. This is known to cause weakness of the tissues in various parts of the body, prominently including the blood vessels and also the intestines. This specific clinical trial is going to test the hypothesis that a specific medication, a treatment, has the ability to prevent or delay the onset of vascular complications. If we can go back one slide. Those vascular complications include what's called a pseudoaneurysm, where blood from inside the blood vessel leaks into an area of weakness directly adjacent to the blood vessel. It also will test the hypothesis that this treatment strategy can prevent a tear along one wall of the blood vessel called a dissection. And perhaps most importantly, will test the hypothesis that this treatment strategy can prevent bleeding outside of a blood vessel, a so-called vascular rupture. Okay, next slide. So in work that largely involved the use of animal models, we were initially able to show that there are multiple cellular events that contribute to vessel wall weakness and ultimately to tear or rupture of blood vessels. Foremost among these was a specific cellular signaling pathway. In these types of pathways, the cell gets cues about what's going on in its environment, what's going on outside the cell. And this leads to a series of chemical events inside the cell that is meant to coordinate a response. Sometimes these cellular signaling pathways and cellular response events are what we call productive. They're protective, they're helpful. But other times, these cellular signaling events are not productive. They're detrimental. They might actually contribute to the progression of disease. In work in vascular EDS mouse models, it was shown that a specific intracellular signaling cascade was contributing to disease. This involved activation of important molecules inside cells, including a molecule called protein kinase C, or PKC, and ultimately another molecule that's called ERK, or ERK. If we advance the slide, we learned that a cascade of signaling down this pathway from PKC to ERK was essential for gradual weakening of blood vessels and ultimately rupture of blood vessels in vascular EDS mice. Using a variety of strategies, we learned that blocking this signaling cascade had the potential to delay, or even more impressively, to prevent vascular events in these mouse models. And that's what led to our effort to find potential medications, to find molecules with drug-like properties that might achieve the goal of inhibiting PKC and ultimately preventing ERK activation in vascular EDS. Fortunately, a number of drug companies had invested a lot of time in developing molecules that have the ability to block PKC activation. One such molecule is called enzastaurin. And when we tried this molecule in vascular EDS mice, we were able to determine that it very effectively blocked PKC activation, prevented ERK activation, and ultimately prevented vascular ruptures from occurring. So that's the basis for this particular trial. Again, it's focused on people with vascular EDS. It's focused on a specific strategy that showed the ability to achieve the goal of preventing vascular events in EDS mouse models. But it's a hypothesis to be tested. We need to develop the evidence in people that this is both efficacious, that it works, that it achieves the goal, and that it's also safe. So all the effort that's being applied now to develop the protocol for this trial relates to designing a study that will efficiently test this idea, and equally importantly, designing a study that will be safe for participants. So I think that's the end of my section.
Thank you so much, Dr. Dietz. And there was a previous webinar that we did with Dr. Dietz and I think a couple other of the amazing doctors on this call talking about the background on that in more depth. So if you're interested in that, I'll throw a link to it at some point in the chat box or include it in the follow-up email that goes out. Thank you so much again, Dr. Dietz. I'm going to pass it now to Dr. Xavier James, who's going to talk about participating in a clinical trial. And Xavier, I've handed you the remote, so I think you're able to control the screen now.
Okay, thanks a lot for giving me the opportunity to speak in this webinar, and also I'm very pleased to be there and discuss with you the importance to participate in a clinical trial for a given patient of any condition, actually, since clinical trials are a real part, a significant part of what we call evidence-based medicine, in the fact that the observation of the efficacy or safety of a given drug cannot be established on one patient only, but on a series of patients, we will see that there are different stages for that to actually establish this observation. So I'm personally a professor of genetics at the Université de Paris in France, and I've been heading the Department of Genetics at the Hospital Européen Georges Pompidou, which is in close to the Eiffel Tower in Paris, and also directing a research group mainly devoted to rare vascular diseases. And we have been working on vascular Ehlers-Danlos syndrome clinically and experimentally for more than 10 years, and actually following approximately more than 100 and 150 patients per year in our center, new patients for some of them, but mainly chronic patients and their families. So we have some experience with VEDS, and as Dr. Aldit said, the need for a medical treatment which is relevant for this condition is obviously of very strong importance. So if I may have the next slide, Kelly, since I'm not using it very properly, probably.
It says that you have it. Click on the screen and try one more time.
I wish it would work.
It looks like it's working now. You click on the screen.
Okay. So, oops.
All right, we'll just stay right here. I got it.
I'm going too fast. Okay, thank you. So what does it mean to participate to a clinical trial? As it is indicated in this slide, there are different phases for clinical research, different stages, if you like, in which scientists or medical doctors are conducting experiments with a health intervention to obtain sufficient evidence for a particular drug to be considered as effective and safe as a medical treatment. So usually phase 1 studies are conducted on small groups of healthy volunteers and are testing the safety of the drug and learn then about side effects. And then we will have from phase 1 study from normal volunteers the knowledge of the not the efficacy, because it's on normal volunteers, but on safety and mainly the main side effect that you can expect. And this kind of phase 1 study have already been performed with enzazosterone with other indoor conditions. The phase 2 trials are conducted then in patients themselves, usually in a series of patients, say 50 approximately patients or 100 patients, depending on the conditions. And the idea there is actually to improve our knowledge on this time on the biological or clinical activity of a drug. And so it's really designed to assess if the drug and how the drugs works. Sometimes it's also used as a dose to determine the dose of the range of dose of the drug. And also, obviously, you will get in this phase 2 trial the idea of the safety of the drug in conditions. It's important to know that obviously the safety will be tested in a time that's usually is approximately the same time that it is going to be done in the phase 3 trial, which is conducted then in a largest group. Usually it's conducted in randomized and controlled way, often multicentral trials, mainly in rare disease like VEDS, and on large patients. Then the idea is to usually compare one arm with a particular drug and the other arm without this particular drug. And the Okay, of
Dr.
the
Jeunemaitre, your
patient, for the
internet,
patients.
I think your internet is cutting out a little bit.
Yeah, maybe. Yeah, I'm sorry for that.
That's okay.
Francis,
ahead.
have you been hearing me at a particular time? I've been cut at the particular time.
Just what you just said prior to when I cut in there.
Okay, so I'm going back to the phase 2 trial or the phase 3 trials.
I think phase 3 is good.
Okay, so I was just saying that the phase 3 trial is usually on a lot of patients, multi-center trials, especially for rare disease. And the idea, sorry that I'm unstable in terms of internet. And the idea is to test the effectiveness and safety of the drug. Then, obviously, for a given patient, enrolling this patient for himself or a member of his family is a strong chance for the doctors to see and to establish the efficacy or the safety of the drug, and also for the patient himself to have the best care as possible. If we can get the same if we can get the next slide, please. I'm sorry for the internet stuff that is not very properly working. What are the benefits of participating in a clinical trial for a given patient? First, it has been very well established in different trials that for the patients themselves to participate to a clinical trial is the best way actually to get the best care at the particular time of the knowledge of this disease. Because the patients are surveyed in a way that everything is really seen in terms of efficacy, safety, and also any event that the disease can cause. Obviously, as I said before, it gives for the patients and the doctors strong knowledge about the efficacy and safety of the drug, not only an acute way, but also a chronic way. What should I know, or what should a patient know before making his decision regarding the clinical trial participation? He should know that all the prerequisites of a particular trial has already been looked at very carefully by what we call an institutional review board, IRB, which is a group of professionals that has been formally designated to review and monitor the corresponding biological research to see if everything was controlled and to protect the patient rights involving the human subject. So really, the idea behind that, it's a very controlled way to test a particular drug and to see if first the trial has been properly designed to obtain a particular objective, and second, to be sure that everything will be recorded and controlled in this particular trial. That was more or less what I wanted to say tonight, but obviously I'll be free to answer to any further questions.
Thank you so much, Dr. Jeunemaitre. I'm going to hand it over now to Dr. Peter Byers, who's going to talk about some of the key terms that you'll hear when we're talking about clinical trials. So Peter, I've just passed you the remote.
That's great. We'll see if it works. And thank you, Katie. And thank you very much for having me here. It's a pleasure to be involved with the group, as it has been for many years. Just a note, I was looking for that shirt. I was also looking for the years that were in that picture, and they seem to have disappeared into the same closet. My job here is really to talk about some of the nomenclature, and Katie, it looks like you're going to have to do it. Oops, I don't think I'm getting. And some of the terms that you'll hear about, that you read about, and that will be part of engaging in this or any other clinical trial that you mind. And the first is the idea of informed consent. And this is something that Xavier referred to in talking about the IRB and what happens. And informed consent is really a process that involves the investigator or the investigator surrogate providing all of the information to you as a subject or to the subjects in the process to consider before participating. And this includes the details about the intent, the purpose of the trial, what's being tested, what the reason for it is, how long it will last, what the endpoints are intended to be, what kinds of tests and procedures you would undergo, how to contact people, and an assessment of what the risks and benefits might be at the beginning, so that you have as much information as you can have at that time. It is actually an interactive process so that you are able to ask questions and to get answers in real time. Usually it's done, it has almost always been done face-to-face, although I can imagine that if we don't clear things up a little bit, that with COVID, that some of this would be done if it's done in that time by meetings such as this one, where you have the opportunity to ask the investigators directly about things. The consent is documented, in which the subject signs a written consent, and that's signed also by the investigator. And then the subject and the investigator both get copies, and it's maintained. And one of the things to emphasize about clinical trials and the process of informed consent is that the participation is voluntary, and the subject can leave the clinical trial at any time without any punishment or any adverse effects being expected. So these are confidential activities, and particularly the information about the participants, the subjects in the trial, may only be disclosed to people who are authorized to see that. And in return, because the trials may involve drugs or activities that are licensed in some way, participants are expected to keep the details of the clinical trial confidential, although many of the details will be available on sites like clinicaltrials.gov if you want to see them. A protocol is a document that contains all of the details about the clinical trial, including its goal, who's eligible to participate, what tests and treatments are involved, and how long the trial will last, and all of the other kinds of information. There's even down to the detail of how many pills you might take and what the timing would be, what the limitations would be. And it's something that you get to keep so that you have that available throughout the time. Trials can be conducted in a number of different ways. In some, which is what the one that Hal was talking about, they can be conducted at multiple centers where the same clinical trial protocol is used at more than one site. So there are many places around the country where there are people who have vascular EDS who have been seen and have been aggregated to different places that would make good sites at which to do them. And that would be a multi-center trial where the rules are all the same for everybody. The selection procedures are the same. The process of informed consent is the same. And all of the medication is the same. And all of the studies are done at the same place and by the same people. I'm sorry, they're done at different sites by different people. There's a centralized trial, and that means that everybody has in-person visits to one of the enrolling sites for drug distribution and other follow-up. And so usually it's all done at one place. And then there's a decentralized trial or remote where it doesn't require travel to a central site. The tools for capturing consent data, monitoring and diagnostics can all be done electronically. There's direct-to-patient drug distribution from a central site. And then the use of local labs and imaging is usually engaged so that there's little or no travel. So blinded doesn't mean that nobody knows what's going on. It means that you still have full information about how the trial is going. It's just that a double-blinded trial means that neither the participant nor the study team knows what treatment the participant is receiving. And this is intended to prevent participants and the researchers from influencing clinical trial results by in some way selecting data or doing something else that would might make one treatment look better than another. A single blind means that one of the parties, either the subject or the investigators, may know, but the best design trials are usually double-blinded. A randomized controlled trial means that the treatments are assigned by chance. That is, essentially, it's like flipping a coin for each person to decide which treatment is being assigned. And in the case of one where there is a treatment arm that has a new drug and a control arm that has standard treatment, the treatment arm would have the new drug and the standard treatment in some cases. The assignment is by using a random number table or by flipping a coin, or the idea is by flipping a coin. Next. Placebo control means that the drug is given, but there's no other drug given to the control group. So there's a placebo, and that maintains the double-blinded status of it, so that the pills, if they're being used, look the same as the ones that contain the treatment medication. The background standard of care, as Hal said, there isn't really a background standard of care, but many people who have vascular EDS are being treated with antihypertensive drugs and in some cases other things like vitamin C. And the intent for background standard of care would be to maintain the ordinary things that people are doing and to add on a investigational medication or to have no additional medications, and then to compare the outcomes in the two groups. One of the critical things that you learn about in the process of getting engaged is that there are inclusion and exclusion criteria, and that is who is being admitted into the trial and would either have the investigational medication or would not. And usually these mean that the people who are included must have the specific condition that's being investigated. And there may be particular kinds of factors that may make a difference. And these would include things like age, gender, type and stage of the condition, and previous treatment history and other conditions, perhaps. These will all be discussed or are all discussed at the time of informed consent. And they're all part of the registration process for determining whether the subject applicants are eligible for the trial. So one of the things that's very important in trials is to know whether something is happening, either whether there's a beneficial effect or an adverse effect. And that means that every event, in fact, every interaction is reviewed by what's called the data monitoring committee, which is an independent committee that's separate from the people who have designed and who are running the trials, to assess the progress and safety of a clinical trial. So they look at any events that might have happened. That is, are there still arterial dissections or arterial ruptures? Are there still other events that are there? And adds them up sequentially as time goes on. And if there is a preponderance of benefit or preponderance of harm that occurs during that time, decisions can be made about whether to continue or modify or stop the trial. So recruitment and enrollment, which are the things that occur at the beginning, is the process of identifying individuals who are suitable to participate in a clinical trial based on the protocol's inclusion and exclusion criteria. So the protocol says, here are the people that we want to study, and the recruitment process engages in that process. And each site for these multi-center sites would have a target number of individuals to enroll in the trial that are dependent on the number of individuals who have relatively easy access to that site and what's known about them. I think that's the end, right? Oh, thanks, Katie.
Yeah, thank you so much, Dr. Byers. I'm going to hand it over to Dr. Shaine Morris now, and we'll try the remote control one last time, but if we strike out, that's okay too.
Okay, that's great. All right, I think I've got it. So thank you for letting me talk. I'm going to speak a little bit about clinical trial analyses and outcome measurements. So this is sort of the math of the trial. And so how do we really measure if a medication that we're trialing is working statistically? And how do we do that rigorously so we can really say there's a difference? For example, say you have 2 classrooms and you're testing the effect of a teaching intervention, and 12 people do well on the test in one classroom of 20 versus 10 people do well in the other classroom of 20. Could that just be chance that 12 did better versus 10, or is that really a difference? But if you have 1,200 people do better versus 1,000 people, that might really be a difference. So the bigger the study, the better. And you really have to use rigor in making these measurements to understand if there's a difference so we can say for sure. So I'm going to go over some of the terms that are being used to really test the hypothesis that a medication in a drug trial or a clinical trial is making a difference. So the first verb, or the first word we're going to talk about, are primary and secondary endpoints. So this is really the outcomes we are looking for in the trial to improve in the treatment group. So these are events, like in vascular EDS, these would be events we're trying to avoid. So endpoints would include, most likely in a study like this, dissections, ruptures, intestinal events, things that we don't want to happen. Now, in any kind of trial, we have to choose a primary endpoint, and that is a thing that we either think is the most important thing to improve or the thing that we think is most likely to improve. And that's the main measure of whether the medication was effective. However, in almost any clinical trial, and including VEDS, there's going to be lots of other endpoints. So you can imagine in VEDS that almost any trial is going to look at ruptures and dissections, but might also, again, look at intestinal events, might look at quality of life, might look at other things and say, was one group different than the other group? So any trial will have a primary endpoint, and then everything else will be a secondary endpoint. The next term to talk about is compliance, and this is in relation to clinical trial. And this is basically sort of talking about what the other speakers talked about, that people running the trial, the physicians and the coordinators, are doing everything according to the rules so that there's not bias and that there's lots of rigor. And again, this allows us to say that the trial was run appropriately and you can believe the results. So this is enrolling people properly, consenting people properly, allowing people to withdraw when they want to withdraw, going exactly by the trial protocol. It's really important because anytime you don't do something in the trial the same way, it could introduce bias. And the results could be as a result of bias, not as a result of the drug treatment actually being better. So it's really important for all sites to be compliant with the protocol and to keep with regulation. This means documenting everything properly, dispensing medications properly, and it's really, really, really critical in a very good clinical trial. The next term is retention. So this is basically keeping patients on the protocol. Now, of course, any person enrolling in a clinical trial can leave at any time without any negative effects. However, the more subjects you have in a trial and the longer they're in a trial, the better you can prove that there's a difference or prove that there's not a difference. And every single person matters and every day that they're in the study matters. So retention is the encouragement and support offered to keep a person in the trial. And so we always account that some people are not going to stay in the trial, but the goal is really to retain as many subjects as possible to really know the truth of whether the drug treatment is working or not working. Should advance. I think it just has a little delay on it. Okay. So next we're going to talk about imaging. So especially in a VEDS trial, imaging is really important. And so imaging are technologies we're using to view the human body to monitor or treat conditions. And so in this, for vascular EDS, we use a variety of imaging modalities. We, of course, commonly use CAT scans and MRIs. Other imaging might be using ultrasound of the extracardiac vessels or echocardiography, which is ultrasound of the heart. And so what you really have in a trial, you have to make sure you're doing similar imaging, because you can imagine if every center could do any imaging they wanted, one center doing imaging once every month and another center doing imaging once every 3 years, they might detect different things. So you have to have similar imaging throughout sites, ideally as surveillance. Now there's surveillance imaging, which is sites in the trial are doing everything at the same in time intervals. Of course, if a patient gets sick and you come into the ER and need imaging done, of course you would get imaging appropriate for that condition. So imaging related to a trial does not preclude any symptomatic imaging. Say a trial said you're going to have imaging every 3 months or every 6 months, and you come in and you're sick, of course you can get imaging then as well. And that imaging would also be included. So there's usually surveillance imaging and of course any acute clinically indicated imaging. And again, this can include, at least for vascular EDS, things like CT, MRI, and ultrasound. So moving on to biostatistics. So biostatistics is basically, like I started to say at the beginning, is math that allows us to say this is truth or not. And is there a difference? There's two things we're looking for in the treatment group and the placebo group or the control group. One, is there a mathematical difference that cannot be due to chance? Could chance have just explained this, or is this a really true difference? And two, is it clinically significant? For example, you might have heard about some cancer studies and they extend length of life 3 weeks or 4 weeks. And there you might say, mathematically, statistically, there's a difference in these two drugs. In this drug, you live 3 weeks longer than in this drug. But is that clinically significant? That's unclear. And so we really design a study to have both statistical significance, so that's math saying it's not just chance, and to have clinical significance, saying this is a difference that would make someone feel better. They would have less events that are measurable. We really are aiming to improve, improvement in the major outcome in a clinically meaningful way. Now, ways we get to statistical significance are sample size. Again, if we had 5 people in each group, there's really not a difference whether you have 1, 2, 3, or 4 people experience negative events, because adverse events, because that's not a big enough group. So the bigger the group is called our sample size, the more power we have, which I'll get to in a second, to calculate a difference. So basically in almost any trial, both the number of patients and the amount of time in the trial help to, the number of patients is the sample size, and then the longer you're in a trial, that helps increase the power. So the power goes with sample size. It's number of participants needed to have a meaningful change. So there has been a power, or there will be a power calculated for any trial, and that will be an estimation of how many people need to be enrolled in each group to tell that there's going to be a difference. So to give an example, a non-clinical trial example, but in a classroom, if you're giving a new intervention, maybe a teaching intervention, and you think that kids are going to do 30% better on the test, you might have to give that intervention to 60 patients in each class to see a difference. And so this is done ahead of any trial. There's a power calculation, and the power calculation says, how many patients do I need in each group, if the difference is as big as we expect, to enroll to see a difference. And so that's what's going on right now in these VEDS trials. And you can imagine, given the rarity of VEDS, this is a challenge, because usually the more patients, the better. You want to have adequate power to show a difference. The other thing that affects power is what percent of patients have an event. If only 1% of patients have an event, you need to have a whole lot more patients. If 50% of patients on average have an event, it's a lot. You can have a lower sample size because you're powered more easily with a small number. So if 50% of patients have an event and you reduce that to 40% or 30%, that's mathematically pretty easy to show. But if you go from 1% to 0.5%, you need a lot of people to show that difference. So this is something that's really, really important in designing these VEDS trials, is getting appropriate power and sample size, which go together. So I think that's all for my section. And I think we're all happy to answer any questions.
Thank you so much, Shaine. So I'm going to go ahead and stop sharing my screen just so that everybody can see the presenters answering the questions. There is a Q&A box. We have a couple questions in there, and there were some questions that were submitted in advance. So I'm going to go ahead and get the ones that are in the Q&A box now, and then head to some of the ones that were submitted in advance. So if anybody has questions, remember this is generally about clinical trials. We can't really answer questions specific, we can answer general questions. So can a participant in a clinical trial find out whether they are receiving the drug or placebo if it's necessary for other medical care? So like an emergency, for example, would that disclosure preclude further participation in the clinical trial? And I don't know who wants to, we didn't discuss how we would divvy out questions. So whoever wants to take it, I think.
I can say that would be a standard safeguard in a clinical trial. If there is an absolute need to know for care of an individial, that information can be shared with their other care providers.
I think that usually does preclude, if you're unblinded, that ends your enrollment in the study typically, right? I mean, I don't know what this study would be, or these two studies coming up. But I think typically once it's unblinded, because that introduces bias. If your providers know what medication you're on, they might treat you differently. So, and I think, depending what medication is trialed, it can be different, important. If it's a novel drug that's trialed, the side effects, you would have to suspect the side effects or the complication you're having is due to that drug. Or if it's a drug that's known, you have you would more likely be suspicious of what side effects to look for. So you'd want to think the complication you're having is associated. So it's something you want to think about carefully, but most trials it's allowed, it can be done.
Thank you. And there's a couple questions in here specific to this trial. I'm going to try to kind of rephrase them in a way that could be answered for clinical trials in general. So I think it was Peter, you had talked about types of disease that might impact enrollment. And can you talk a little bit about how that might impact like a group of people with VEDS being considered for enrollment for a trial? Like, is there a specific type of mutation that might be more desirable for a trial, or?
Sure, and I think that a number of us can talk about this. But we know, for example, that in vascular EDS that people who are heterozygous for null mutations or who have haploinsufficiency, that is one copy of the gene just has gone to sleep, but it doesn't produce an abnormal protein, have a very different trajectory to their when complications start and what their ultimate survival would be without treatment, which is really quite different than what happens with people who have the more common substitutions for glycine or the exon skipping mutations. That means if they're included, then there's a set of people that over a relatively short time is probably not going to produce a signal. We can't anticipate that they would have events, and so because they don't have events, it's not really attributable to the drug in most instances. So if we are looking at a relatively short time for getting an outcome, we'd like to have people who are predicted to have events during the period of the trial. And that would mean that we would leave those people out for the most part. Not because we don't think they deserve treatment, but because we would be looking for treatments that probably act differently or may act differently.
And would that be the same for somebody with a mosaic mutation?
Yeah, mosaic. At this point, I don't think we've identified, I'm not sure about how Xavier or Shaine, any people who are symptomatic and symptomatic. So you can imagine that there would be instances where there might be if there are one or more blood vessels that have the abnormal clone and make one, a region of that blood vessel susceptible to being damaged by the disease process. But for the most part, as far as we can tell, individuals who are mosaic for a specific mutation do not have a change either in the frequency of events or do not have the events that are usually associated with vascular EDS. And that's because when you're mosaic, each cell is different, and some may have the mutation, some don't. And so the effect is very local, as opposed to with vascular EDS, where every cell has the mutation. And so it's making stuff that goes around it and having a very broad effect on the tissues that are involved. So I think that we are unlikely to include people who are mosaic as well. And that would be true for many studies where the mosaic individual is not affected. Now, it's quite different than, there's a category of neurologic diseases, particularly where only people who are mosaic who are identified, because people who have full expression of the gene in all cells don't survive.
Thank you. And I think, Shaine, I cut you off. Were you going to say something else?
Oh, no, that's fine.
Okay. Okay, so let's, I'm going to move on to a question that came in on the ahead of time. And I think I'm going to open this to Dr. Dietz and Dr. Jeunemaitre. Maybe one of you can go first and the other one can add in if necessary. There's a broad question about what have clinical trials for VEDS or Marfans taught us up to this point? And I think that really just, what is the significance of trials in the past and where are we?
Maybe I begin and then Harl will complete my answer. This is a very important question, actually, since trials, or at least double-blind trials in these rare conditions have been rare themselves. And so I would say that there are two lessons that have been given for the moment. The first one in Marfan syndrome is for the moment saying that, as Harl did said, the mouse model is not always exactly behaving as a human model is, or if a human model can be said like that, since humans are in general much more heterogeneous than a mouse line can be. And so in Marfan syndrome, the very strong beneficial effect of ARBs, which is a particular class of drugs, have not been completely replicated in humans, and probably Al could complete on that. The second lesson is that in VEDS, there has been an open trial performed in Europe 20 years ago now on a particular beta blocker, which seems on a small portion of a small number of patients to be efficient. And then again, the statistics that Shaine was talking about is very important, since this trial, which was interesting, was performed on a relatively low number of patients. And so the generality of the results are maybe questionable. So this is of importance in terms of how to design the trials, in terms of statistical power, in terms of length of the trials itself, and in terms of observation of the main endpoints, primary endpoints or secondary endpoints. So all of these observation and trials that have been performed before, I think, are very important to design the best control trial as possible for this new trial now. Al, could you complete?
Sure. So I agree with everything Xavier said. I think the lesson is that clinical trials are difficult. Clinical trials do not necessarily lead to truth. They answer the question that you ask. So you have to be very careful in crafting the initial question that you intend to answer. If we take the experience of ARBs in Marfan syndrome, the initial trial with best intent asked whether a standard dose of a drug called losartan was as good as a very high dose of atenolol, or better. And the answer that we got from that trial was that they were pretty much just as good as each other, but one wasn't better. There have been many subsequent trials with refinement of the protocol, the question being asked, how the question is being answered. And what's gradually occurred over time is a more clear signal that ARBs are beneficial for people with Marfan syndrome, but it's taken 7 studies and 1,500 participants to get to a more clear answer. What we're attempting to do now, but what this good group is really struggling to do, is to ask and answer the best question right from the start. We are going to do our best to design a very efficient trial that has the capacity to answer the question of, does this drug work or not? Is this drug safe or not? But there may be need for further refinement over time. I think that there are, as people know, other trials anticipated and being planned. Some have different study designs from the other. One is not necessarily better than the other, but I think that it's important for people to feel empowered to ask their doctors and to ask the people running the trials tell me what's the benefit of being in this trial? How does the question that you're asking and answering, how does that differ? And, when, if the results are not what we hope for, what is anticipated as the next step forward? This is clearly an evolution in learning. And, I think it's also important to know that we all have the same common goal of improving the length and quality of life for people with vascular EDS. So we're all hoping that that everyone wins, that there are positive answers and great results from all the trials.
Thank you so much. I'm going to go to the next question, and I'm going to hand this to Shaine. I think you've been typing an answer in there.
Oh, I started, but I realized you were reading it live. So
yeah, I think it's a great question to answer live in case anybody else has it. So when we're talking about events, you're presenting and talking about statistical significance and clinical significance. Could you elaborate? Is the goal to try to trigger an event, or what did you mean by?
Sure. So absolutely not. The goal is definitely not to trigger an event, but the goal would be that the control group or the placebo group, so we're just observing the two groups, but one group got the medication. So if both were placebo, they would just be that people have events at the same rate that we observe in the population now. The goal of the new medication is to reduce events in the arm that's getting the medical treatment. And so we want to see we hope to see, if the drug is effective, a reduction in events in the intervention arm and the same rate of events. I mean, it would be great if both arms had lower events, but what statistically, if we think it's reducing events, you would see less events in the arm that gets medication, and the control arm would have the same number of events we would expect even if the trial was not occurring. So say that 20% of people would have an event in the next 5 years. You might see that in one arm, but the goal would be to have maybe 10% or 5% in the arm that's getting an intervention. So it's just a reduction in events in the treatment arm is what we're looking for.
Thank you.
Sure.
And I'm going to hand the next question, I think, to Xavier. So this is a question that came in ahead of time. It's a specific question. I'm going to generalize it for the purpose of just understanding clinical trials overall. When can trials be conducted in more than one country, and how is it determined, like what countries can participate in the trial, or what people from different countries, whether they can participate? We're getting a lot of questions from specific countries from people who want to be involved and they just don't know if they can.
Sure. I think it's very important for especially a rare disease to have an international or at least a multi-center participation to allow, as Shaine pointed out, the statistical power which is necessary for achieving an observation which might be positive or negative, at least to have a true answer to the question or the hypothesis that we make. So in that way, as there is an international network or international centers which are really focusing on VEDS, I think it's a very nice opportunity through this trial to have a large number of VEDS patients participating to the trial. Obviously, the regulatory conditions from one country to another one may vary, and we have to take care of that. But it's not an obstacle which is insurmountable, I would say, since there are many different trials on other drugs, on other type of disease, chronic disease, for example, where you have an international collaboration on that. So we have to take care of this problem, which is not a problem, but a difficulty where you have usually a company which leads, or a particular center or principal investigator which is leading the trial, and then where you have different countries which are involved in that. with regulatory conditions, which are sometimes specific, but usually general for this trial. I don't know if Al or Shaine want to add something on that.
Okay.
I think that's good.
Thank you. All right, I'm going to jump to another question. I'm going to jump over to Dr. Byers. So this is another one that came in ahead of time. And Dr. Byers, you presented this section about, what's expected of a participant in a trial and, maybe how often they check in and things like that. Can you elaborate on what's expected of somebody in a clinical trial usually? And, are there other things that they need to be aware of during that time?
The expectations for clinical trials will differ among trials. Some expect people to be seen on a relatively infrequent basis, to have all reporting done by electronically or through a website or something like that. There are others that will need to have observations done that may include imaging of different kinds, as Shaine indicated. So they're really very specific. The expectations for the subject, for the people involved, are quite different depending on what the objectives are of the trial and what the outcomes are that are being measured. So it's important when considering engaging in a trial to look at what those expectations are and to see whether they fit the kinds of things that you're willing to do and you're interested in doing, and use that as the basis for the decision about whether to become involved or not. I think, are there things that might happen in the course of the trial? Well, I think we know that there are people that can have the kinds of complications that we see in people with vascular EDS, and the expectation is that they will be treated in the way in which is in the best interest of the participant, and that person changes from a subject then to a patient in the local environment where the person is, and the hope is that unless that event is something I think Xavier just pointed out, is the key to making decisions about whether to continue or not continue the trial, that person would remain in the trial.
Thank you. I think that answered another question that came in that we talked about earlier as well, having an event and maybe, needing to disclose whether or not or find out whether or not you're on the placebo or medication. And, Shaine, correct me if I'm wrong, but I think the answer to that would be that if it's unblinded, that person would then be unenrolled, out of the trial from that point on.
In most studies, yes, but the data up until then is still gets to be used. It's just then their enrollment would end.
Great, thank you. Anybody who wants to grab this one can grab this question. How does the oh man, I'm stumped here on how to ask this question. So let me start over and maybe not use the word how. Can trials include patients that are on certain medications? Are there trials that involve people staying on the medications that they're currently on? And what does that look like in a clinical trial?
Absolutely. I'm happy to start and
Okay.
anyone can
Go ahead.
jump in. But absolutely, it depends how the trial is set up. But for example, the most recent Marfan trials, Hal alluded that some of the Marfan trials have changed recently. And what a lot of them have done is have patients stay on what there is considered standard of care, and beta blockers for many patients with Marfan syndrome are considered standard of care. And so they stay on what they're on, and then they can have an added therapy to their standard of care regimen or not have an added therapy to their standard of care regimen. So for example, the AIMS trial was where irbesartan was added to standard of care, and a huge proportion of those patients were already on beta blockers. And so really it ended up testing, is combination therapy or something added to your standard of care okay? And I can anticipate that in vascular EDS, after Shireen did a survey that most people would not want to alter what their standard of care is, that any study or certain studies would allow you to stay on what you're already on, as long as that's considered sort of within realms of standard of care. Does that make sense?
Yes, I think so. And I'm sorry for anybody who feels like I'm being cryptic here. You know, we are answering questions broadly about understanding clinical trials. So if you're submitting questions about a specific trial, we're trying to broaden those questions and answer them on a higher level, because the protocols for these trials are still being developed. And so some of the specific answers that you're after just aren't available yet. But you know, they will be eventually. And we look forward to that very much.
Kate, if I could just add something to the last question you asked. So if someone is receiving a medication that might have a negative interaction with the study drug, you know, there might be some sort of, for example, a higher risk of a complication, then it's possible that that would lead to either the necessity to stop that medication if that's possible, or to the lack of eligibility of someone to be enrolled in the study. So again, safety is a guiding principle in any clinical trial, and it may not be wise for people taking some medications to enter a trial.
Thank you for elaborating on that.
Yeah, I think one of the things to remember that a trial is a trial, that we don't know what the expected outcome is, but it is not our expectation that things would be worse, or that we would change the way that people are behaving in a standard fashion to in some way make their care less good, simply for the basis of control. So I think, you know, the intent of good trials, which we hope all of these will be, is absolutely to not harm people, and hopefully to do that, to find benefits.
Thank you. There's a couple questions that came in ahead of time about children and trials. Somebody answer, you know, what determines whether or not a trial can include children in it?
So, you know, once again, safety is a guiding principle. In some trials, a medication is being used that's known to be safe in children. That might lead to a earlier or more liberal decision to enroll children in a clinical trial. If a drug has not been used previously for a different indication, or if there's limited experience in childhood, there's often a decision to first gain experience showing safety in older individuals before generalizing the trial, you know, or expanding the trial to include children. People could probably recognize that pattern for what's happened in the rollout of COVID vaccination, for example. So the guiding principle is safety, and one piece of leverage that we can use is prior experience with that drug or class of drugs in the pediatric population.
If I could add to what Hal said, one thing that unfortunately has happened in the history of trials, especially in America and the United States, is that in terms of safety, people have excluded children from most trials, which actually makes it less safe for children, because people end up using the medications in children and we have no idea if it's efficacious or safe. And so there's been a big movement, people consider it sort of discrimination against children. So there's a big movement and to make sure that children are part of trials in some way. It might be staged, as Dr. Dietz said, and as we saw with the COVID vaccine, but there's a big push to have children included as often as possible so that we know if it's safe or efficacious in children to get these medications.
If I just may add a word on that, I think there are two things which are important. First, sometimes there are regulation or regulatory conditions which might be specific to children. And second, the choice to involve children is also dependent on the frequency of events that you can expect depending on the condition that you're using, the condition that you're analyzing. Say, for example, for VDS, that you would involve very young children at the age of 5 or 10, expecting in the 2 years of a trial to have a certain percentage of vascular events would be probably very difficult, since we know by experience that young children usually do not develop arterial events in their youth. So that's why we have to think about the proper age to involve young patients and to first be beneficial for them, and also in terms of statistic to be sufficiently powerful to get a possible difference between the active drug compared to the placebo control.
Right. It's sort of that balance between power, it's sort of just like the COVID studies. They predominantly, in the COVID vaccine studies, they predominantly enrolled people who had a high exposure. So you would really have a contrast between those that got infected and those that got the vaccine and hopefully did not get infected. It's the same issue with vascular EDS, is you really want to show a contrast between your groups to show a reduction in events statistically, to show a difference. And so if your control group is not expected to have events, then it's not as helpful to have that person enrolled and go through all these rigorous evaluations and imaging.
Thank you. And I know that that's a question like on everybody's mind, is whether or not these trials will include children. So I really appreciate the elaboration on what goes into that decision in the first place. I think that's helpful for people to understand. There's a question in here that I think is really interesting. And sorry, there's a cat here trying to get on my lap. So if there are two trials that coincide or happen at the same time, that are separate trials, is there like a likelihood that people could be in both, or what would determine whether or not somebody can be in both trials or only ones? How is that decision made?
Well, if I just may answer on that, on the French part of it, I suppose it's the same thing in other countries. It's completely forbidden to be involved in two trials at the same time. Because you obviously would bias any results, and in addition to that, you could also have a side effect that would be unexpected if you don't know that the patient himself is on a particular drug or placebo on another trial. And in addition to that, probably you would have other type of exams, which are usually already done or performed in a particular trial. So in France, it's impossible to participate to two trials at the same time.
I think there's a strong argument to be made to avoid people being enrolled in two trials simultaneously, even if there is not specific regulation that precludes it. It just introduces too many uncontrolled variables. It could completely throw off power calculations. There could be safety ramifications. So I think it's very unlikely and close to impossible that people would be allowed to enroll in two clinical trials simultaneously.
If you can imagine that if the trial were not blinded, that it would be possible to have a trial in which the control group was the same for two, as long as the selection criteria were the same for the trial. And that people could if they were randomly allocated to a control group in group 1, that they could be considered a control group for the second population.
Yeah, but it wouldn't be random then. So then
It wouldn't, well, it would be random.
for one of the studies, it wouldn't be random.
Right, right, exactly. You'd have to have dual randomness to.
Yeah. Well, some people do that. They have if you have a big enough population, they'll have
Yeah.
3 groups, right?
Yeah.
So
Yeah.
they'll have a control intervention A and intervention B. And
Right.
that was, definitely, I don't know for this study of Stern coming up, but for certain beta blocker protocols that have been toyed with in the past have considered that, but the VEDS population is so small that having 3 intervention group is probably not feasible statistically.
But we may find out how big the vascular Dietz group is as part of this.
Yeah.
And it may turn out to be quite a bit bigger than we think it is.
Yeah, maybe in the future it might be, maybe follow-up studies could be bigger too.
Yeah.
Thank you so much for answering that question. I think that's a big one with, a couple trials coming up. The big question is, can you be in both of them? And I think, hopefully this webinar will provide a bit of background and understanding on all of the things that go into clinical trials so that when you look at both of them, you can decide as an individual if you want to be involved and which one appeals to you. Having some kind of background knowledge going into it, I think that's important. And that's part of why we wanted to do this webinar, to make sure that everybody really has a good understanding of clinical trials. I think that will probably be maybe one final question. What is the timing for, all of these questions I've asked you, I've like generalized and said we can't provide specific answers. Is there a timing for finalizing study designs for these trials? And, what do we expect? Like, what can we look forward to in the future? Like, what is a timeline that we can expect to be able to answer these questions? Is that answerable?
I would say there's high incentive among all parties to have a responsible, inclusive trial launched as soon as feasible, given the amount of preparation that has to occur. So and I would imagine that's true for both trials that are anticipated. There's high incentive, but I don't think that there's additional specific information to share at this time.
But Hal, maybe you could talk a little bit about what the process is. How do you get to the point of being able to get, what are the steps that are involved? And that may give people an idea about what needs to be fulfilled and how it works. Yeah,
so there are regulatory paths that need to be followed. There is, for example, a general application procedure in the United States to the Food and Drug Administration to register a new prospective therapy, for example, a new drug as an investigational new drug. And that allows the specific planning process for the trial to proceed. Ultimately, there are, again, federal regulatory agencies that will review the protocol and make sure that it, adheres to the principle, the guiding principle of safety, and also has a reasonable chance of testing the hypothesis of efficacy. Participating sites would need to be identified and would need to prove the capability of carrying out the procedures of the trial. As was mentioned by prior speakers, there is the need to also develop regulatory committees for the trial that are independent from the sites conducting the trial. The Safety Monitoring Board and the committee that's charged with doing the math, the biostatistics that Shaine mentioned, to test the hypothesis of efficacy and to assure safety as the trial is being conducted. The important process of patient recruitment would then need to begin, which also includes an essential component of patient education. So people are prepared to ask all the important questions during the informed consent process. Ultimately, the trial would then be launched, and there would be a protocol that would dictate how often people would be seen and what monitoring studies would be performed. The efficacy and safety committee would be meeting at fixed intervals to assure that things are proceeding as specified in the protocol, and to review the data as they're coming in. Ultimately, it is anticipated that it would be years of data collection that would be involved in a trial such as those that are being anticipated. And ultimately, once definitive evidence was available in either direction, there would be some form of an announcement, typically a publication that would detail the results of the trial. It's the process, the regulatory process takes typically months for trials, sometimes even a year or more. The patient recruitment is anticipated to take many months to years. Every patient that's enrolled is anticipated to be followed for years in the trials that are anticipated. So we are talking about a process that will play out over an extended period of time.
Thank you so much. And I think that concludes our Q&A today and our webinar. I do have a couple more screens to share before I let everybody go tonight. First off, thank you so much to all of the presenters tonight for taking the time out of your day and for just your dedication as community. Extra kudos to Dr. Jeunemaitre. I think it's like 2:30 in the morning there now. So thank you so much for joining us from France. We really do appreciate it. This webinar was recorded, so there will be a link to the recording that goes out in the next couple of days. And if you're looking for specific information about these trials that are coming up or being planned or developed, the PREVENT trial, which is the ensifentrine trial, you can get information at preventvedstrial.com. I think you just put in your name and your email address and stuff, and then you'll get updates as they come out. So as Hal said, it can take some time for these things to happen, but if you're signed up there, you should be getting updates as soon as they are announced. And we also will be providing updates, as they come out as well through the VEDS movement. The Discover trial, which is the celiprolol trial, also has a website, discoverceliprolol.com. Very similarly, you put in your information and then you will get updates as they are announced. So these are two really good tools that are available to you if you're on the edge of your seat like I am. This is a great thing to go ahead and take the time to do. And finally, this is a natural history study that has already been enrolling for some time now, the VEDS Collaborative Natural History Study. It's available through the University of Washington. If you're on this webinar and you haven't looked at that or haven't considered enrolling in that, you can visit the link here. I'm not going to say the entire link, but it's here on the screen and you can copy paste it That is a natural history study to just look at, people with VEDS as a whole. So not the kind of clinical trial that we've been talking about tonight, but still very, very important as research continues in vascular EDS. And if you have a question that was not answered tonight, that can be answered if you've submitted specific medical questions or are looking for more support, I encourage you to reach out to our Help and Resource Center. This is a picture of Jan Lynch. She's the nurse who runs our Help and Resource Center. She is fantastic. You can visit thevedsmovement.org/ask and submit your questions there. And that is it for tonight. Thank you so much again, everybody, and we will see you soon.
Bonne nuit.
You.
Bye, everyone.
Bye.
Thank you so much.